Okay, so let's do labs 37 and 38. This is the presentation. So here you can see the learning objectives of what you're going to grasp by watching this presentation or through the assignments that I've given you through Connect, okay? So let's get started. So here I want to talk about the characteristic of blood. So blood is a type of connective tissue suspended in a liquid matrix known as plasma, okay? Blood is very important because this is going to deal with transporting vital substances such as gases and nutrients. It helps with maintaining stability of our interstitial fluid and distributing body heat. And the amount of blood will actually vary with body size, changes in fluid concentration, changes in electrolyte concentration, and the amount of adipose tissue. Okay. Blood is about 88, sorry, not 88, sorry. Blood is about 8% of body weight. The adult blood volume is about 4 to 5 liters in a female and 5 to 6 liters in a male. Okay. So we have blood cells, as you can see here, and they form mostly within the red bone marrow and are called form elements. Okay. So red blood cells, I'll definitely abbreviate those as RBCs. White blood cells, you might see me abbreviate those as WBCs, and platelets are also known as cell fragments. So if you hear me say cell fragment, I can use those interchangeably. So here, this is once you've centrifuged a blood sample, you basically put this in this machine that spins around really fast, and it separates the composition of blood based on the different weights of the liquid material. So here you can see the total volume of blood. And so the percentage of red blood cells is called a hematocrit. Sometimes you might see it abbreviated HCT, or also known as a PCV, known as packed cell volume. And 55% of it is going to be plasma, 45% is going to be red blood cells. And then the white blood cells and platelets or cell fragments typically is less than 1%. Okay. So this is a chart explaining the composition of blood. If you have any issues in looking at these images, you can definitely download this presentation and enlarge it, okay? Just in case you're having an issue seeing it, okay? So this is the different composition of blood and the percentages, okay? You definitely want to make sure you understand how the composition of blood and the percentages play a role. So let's talk about red blood cells. So red blood cells are also known as erythrocytes. They are going to be, the structure is going to be like this biconcave disc, and 1/3 of it's going to be hemoglobin. So if you see O2 for a red blood cell, that means that's oxyhemoglobin. And if you see without oxygen, like it's deoxygenated, then that's known as deoxyhemoglobin. So that's without O2. So red blood cells or RBCs, they actually will lack a nuclei and a mitochondria. It gets discarded through development, and they cannot divide, but they can produce ATP. They end up producing that through glycolysis. So there's different types of white blood cells. There's 5 types. We have granulocytes and agranulocytes. Granulocytes are going to contain granules, and they have a very short lifespan, which as agranulocytes, they really don't have these granules. Okay. Sorry. So these are the 3 white blood cells that are granulocytes. They are the neutrophils, eosinophils, basophils, lymphocytes, and monocytes. And so they're going to protect, all of these white blood cells are going to protect against diseases. Leukocytes have a limited lifespan, so they must constantly be replaced. And white blood cells are going to be produced in the red bone marrow under the control of hormones such as interleukins and colony stimulating factors. Okay. So let's first talk about the neutrophils. So the neutrophil, these are just white blood cells surrounding the neutrophil. So the neutrophil is going to be this small light purple granule in the acid base stain. So you can see that it usually has between 2 to 5 lobes, also known as PMS. That stands for polymorph nuclear leukocytes. Okay, that's what it stands for. And these are first to arrive at infection sites. So these are like our first line of defense. They have they're very strong phagocytes, meaning that these are cells that's going to engulf foreign material. And they're typically going to make up like 54 to 62% of the leukocyte population. You will definitely see these elevated in your lab results if you have a bacteria infection. The eosinophils, these are going to have these very coarse granules, as you can see right here, and it's stained deep red in the acid stain. You can see that it's a bilobe nucleus, and they are very moderate in the regards of a response to allergic reaction. And this is really going to defend against parasitic worm infections. Okay. This makes up between 1 to 3% of our leukocytes population, and these will definitely be elevated in a parasitic worm infestation or allergic reaction, okay, in your lab results. The basophils, these are going to contain these large granules and is stained deep blue in basic stain. The granules can obscure the view of the nucleus sometimes because they're so large. And the basophils will actually release histamine to stimulate inflammation, and it will also release heparin to stop blood from clotting. Okay, this will actually makes up less than 1% of the leukocyte population, and it's very similar to the eosinophils in size and shape of nuclei. Okay. Then we have the monocytes. So the monocytes is going to be the largest of the white blood cells. This is very spherical, and it has this kidney shape or oval lobe nuclei. This is definitely an agranulocyte. It leaves the bloodstream to become a macrophage. It has that capability of doing that. And this is going to make up around 3 to 9% of the leukocyte population. And this will actually stay alive for weeks/months. Okay, these cells don't die as fast, and they will also deal with phagocytizing bacteria, disc cells, or other extracellular debris. And then we have the lymphocytes, and the lymphocytes are slightly larger than the red blood cells, but they're small. It's the smallest of the white blood cells in regards of size. You're going to see this large spherical nucleus surrounded by a thin rim of cytoplasm, and this is an agranulocyte. T cells and B cells are major types of lymphocytes because they both are important in our immunity, in our immune system. And T cells are a type of lymphocyte that will directly attack pathogens or tumor cells, whereas B cells of the lymphocytes will actually produce antibodies to attack foreign substances. Lymphocytes will actually make up 25 to 33% of the leukocyte population, and they will actually live for years. Okay, these cells can live for years. Okay, they have a nice long lifespan. So let's talk about the general functions of the white blood cell. So here you see this image, and you see a cell, white blood cell, squeezing between the cell of a capillary wall, and it's leaving the blood vessel to migrate towards the site of infection. That's what's known as diapetus, okay? Then there's some other types of functions of white blood cells, such as cellular adhesion molecules. So basically, your white blood cells will secrete proteins that will direct the leukocytes to the site of injury. And then, like I said earlier, your white blood cells have the function to phagocytize or phagocytosis, meaning engulfing and digesting pathogens. Really going to be those neutrophils and monocytes, because those are going to be the most mobile and active phagocytes. Then your white blood cells will be able to have an inflammatory response. So reactions that restrict the spread of infection it's going to really be promoted by basophils, by secreting that heparin and histamine. That is going to basically involve or induce swelling and increase capillary permeability. Okay. And then there's this term known as positive chemotaxis. And basically, chemotaxis is like a chemical trail pathway. I don't know if anybody is familiar with the story of Hansel and Gretel. I think it's Hansel and Gretel, but basically, it's been so long, but I think it's the Hansel and Gretel where basically they leave like either cookie trails or candy trails to get them out the wilderness. I think it's Hansel and Gretel. So anyway, the concept is the same thing with positive chemotaxis. So what happens is the white blood cells will be attracted to the infection site. But the way that it does that is that the cell that got damaged at the infection site will actually leave a trail of chemicals. Okay. And that's released by those damaged cells. And just like how you like left the cookie crumbs to get out of the wilderness during Hansel and Gretel, I think it's Hansel and Gretel. Those chemicals will leave a trail for the white blood cells and it will attract them and it'll lead them to the infection site. And that's what's known as positive chemotaxis. Then another thing that I wanted to say is that neutrophils will respond to a bacterial invasion by accumulating in the infection site and destroying the pathogen by phagocytosis. So sometimes when you have that accumulation of dead foreign cells, that's what can form pus. Okay, that's what pus is, or like when you pop a pimple and all that stuff, that's just basically the dead cells that was getting killed or fight off during our immune response. What I want to just say here is just talk about the white blood cell count. So basically, there's a high white blood cell count and a low white blood cell count. These are the numbers that constitutes that. But you can sometimes have a high white blood cell count due to either vigorous exercising or a great loss of body fluids. Or these are the symptoms that can, or the diseases that can cause a low white blood cells, leukopenia, leukocytosis. So basically like chickenpox, AIDS, anemia, all those things are what constitutes a low white blood cell count. And a differential white blood cell count is basically just the percentages of the different types of leukocytes. And so that can change depending on which disease or infection you're dealing with. Okay. I tend to really explain my images and not go into great detail of slides that already have words on it because you can definitely need them. Okay. Just FYI. Now let's talk about platelets. So platelets are basically these cytoplasmic fragments of a megakaryocyte. And so a megakaryocyte is like this big, big, just humongous thrombocyte. And it's formed within the stem cell of the red bone marrow. As you can see here, this little pathway. And what happens is megakaryocyte will actually be produced by, it's just fragments. I mean, sorry, not mega. Platelets or cell fragments are produced from the megakaryocyte. Like it just breaks off, it just breaks apart. And the importance of the platelets is to help in hemostasis. And hemostasis is basically stoppage of bleeding in damaged blood vessels. So what happens is those platelets will stick to the broken surface and it will release serotonin, which actually causes the smooth muscles in the walls of the broken blood vessels to contract. And it helps with clogging up. It's going to help with clogging up the injured sites so you can stop bleeding. Okay. And so here is a chart explaining that. You can definitely look at that, read it on your own. And now what I want to talk about is the different blood types, okay? So so in 1910, right? So what? That's like over 100 years ago now. The identification of the ABO blood antigen gene explained the observed blood type incompatibilities. Okay, So blood types are distinguished by proteins on the surface of red blood cells. And these can be determined by the proteins or by the genes that encode them. So let's first do some identification so you can understand this image, right? I'm gonna make this a little bit bigger. So an antigen, as you can see, there's antigen A, antigen B. An antigen is any molecule that invokes an immune response. Okay. So if the immune system finds a foreign antigen in the body, it produces antibodies against the antigen. Then there's antibodies, as you can see here. Antibodies are proteins that react against a specific antigen. In an incompatible blood transfusion, what happens is a donor red blood cells will evoke an immune response in the recipient, and those antibodies in the recipient's plasma will agglutinate the donor's red blood cells. So you're like, what the heck is agglutinate? So agglutination means the clumping of red blood cells, which occurs when an antibody in the recipient's plasma encounters its specific antigen, or the you could say the donor's RBCs. There are 33 known antigens on a red blood cell membrane, but only a few can actually evoke a serious transfusion reaction. So only the antigens of the ABOs and the RH groups will evoke a serious transfusion reaction. And so that's why we're learning them. Okay. So here you can see that the ABO blood groups is based on the presence or absence of two major antigens on a red blood cell membrane. You have antigen A and antigen B. Antigen A and antigen B are carbohydrates. And so antibodies are associated with some blood types. So in general, a person produces antibodies against antigens that are not present on his/her red blood cell membrane. Does that make sense, guys? I'm only saying it like this because it can be confusing at first. So I'm trying my best to make sure. And if you need to repeat and listen to this video over and over and write and take notes as you're doing so, do it, okay? Because if you can organize your thoughts, it'll help you learn and study better. So as you can see, for somebody who has type A blood, you can see they have antigen A on their surface of their red blood cell, but they have anti-B antibodies. Okay, that's what I just explained. So like I said earlier, agglutination is basically when a red blood cell comes in contact with antibodies against them, and they will agglutinate and clump together. Okay. And so here is basically the compatible blood types for transfusions. This is a chart for it. So type O is known as the universal donor. And so type O will actually lack the A and B antigen. So type O red blood cells could be donated to a person with any blood type. Often when given to a person with a different blood type, only the red blood cells are donated, since the plasma will actually contain the antibodies. So when you're donating blood, you're not giving them the plasma and the red blood cells. You're just giving them the red blood cells. And we already showed you how you can separate that. And remember, the antibodies are located within the plasma only. Then there's type AB blood, and that's known as the universal recipient. So type AB blood lacks both anti-A and anti-B antibodies. So an individual with type AB blood can receive donor RBs of any type. But wait, there's more. So there's this thing known as RH compatibility, or also known as the rhesus factor. The rhesus factor, or RH, is also known as antigen D, D as in dog. And basically, this is where the positive and negatives comes in, okay? So the RH group was named for the rhesus monkey in which it was first studied, okay? And so rhesus is spelled RHESUS, by the way. So the group includes several RH antigens or factors, but most important one is the antigen D. Okay, that's the most important one that we're going to be looking at in regards of the Rh factor. So when somebody says they're Rh positive, that means that the presence of antigen D or other Rh antigens on the red blood cell membrane is there. But if somebody said they're Rh negative, that means that they do not have the Rh antigen on the red blood cell membrane. Okay. And so anti-Rh antibodies form only in Rh negative individuals in response to the presence of red blood cells with Rh antigens. Okay. And so the seriousness of the Rh blood group is evident in a fetus that develops in the condition known as hemolytic disease of the newborn. Okay. So basically, in this image right here, you can see that the mom is Rh negative with a fetus that is Rh positive. Well, during childbirth, the cells from the Rh positive fetus may enter the mother's bloodstream. So what happens is the mother becomes sensitized and antibodies are formed against the Rh positive blood cell between the pregnancy. So if she was to have a second child and in the next Rh positive pregnancy, so I don't know why I said presidency, but pregnancy, the maternal or the mother's antibodies that's now formed will actually attack the fetus red blood cell. That's what's known as the hemolytic disease of the newborn. And so what happens is if that happens, we typically give the mother Rogam. So it's like an immunosuppressant drug to prevent this from happening. Okay. So that's what it means to have Rh factor. And so here, this is a video. You definitely have to download it and then hit play in order to view it, okay? Of explaining the blood types and the Rh factors, okay? If you want more resources. Now I want to talk about lab 38. So here's the learning objectives and let's get started. So the heart is going to pump around 7,000 liters of blood through the body each day, and the heart contracts around 2.5 billion times in an average lifetime. Okay. So the heart and all of the blood vessels are going to make up the cardiovascular system. And so the left side of the heart is going to contain oxygen rich blood, while the right side of the heart is going to contain oxygen poor blood. Okay. So that's why you can see it's color coordinated, blue and red, poor with blue, rich with red. Blood is going to run through two circuits. That's what's known as the pulmonary circuit, which is going to go to the lungs, and the systemic circuit, which actually will go to the cells of the body. Okay, so when you hear the word cardiovascular, that actually refers to both the heart and blood vessels. And so the pumping action of the heart transports blood through the blood vessels, and the blood vessels form two circuits. And so that's two circuits that I said earlier. First, the pulmonary circuit, that's going to transport oxygen poor blood from the heart to the lungs and back to the heart. In the lungs, the blood picks up oxygen and drops off carbon dioxide. For the systemic circuit, it's going to transport oxygen-rich blood from the heart to all body cells and back to the heart. And so blood is going to deliver nutrients to the cells and remove the wastes, like move the waste back to the heart. So here you can see the size and location of the heart. So the heart is this hollow cone-shaped muscular pump. In the mediastinum, this is where it's going to be located within the thoracic cavity, superior to the diaphragm. And the heart contains four chambers. We have two upper chambers and two lower chambers. Those are known as the two atria and the two ventricles. Atria is plural, atrium is singular. Okay. The lower chambers is really where we're going to get our pumping action from. So the heart size will actually vary with body size. And so the heart is going to lie within the thoracic cavity. and it's going to be an average of 14 centimeters long and 9 centimeters wide. Okay. So, like I said, the location posterior to the sternum, medial to the lungs, and it's going to be anterior to the vertebral column. Okay. Um, at the base, okay, at the base of the heart is going to lie beneath the second rib. Sorry, and the apex is down here. The base of the heart is right here. So the base is going to lie beneath the second rib, whereas the apex is going to lie at the fifth intercostal space. Okay. And so then there's coverings of the heart. So let's talk about the coverings of the heart. So we have this term known as the pericardium. And the pericardium is covering over the heart and large blood vessels. So we have the fibrous pericardium, the parietal pericardium, and the visceral pericardium. Okay. The visceral pericardium, just for the record, is also known as the epicardium. Just when we talk about the walls of the heart, just know that the visceral pericardium and the epicardium are used interchangeably because it's the same thing. So the fibrous pericardium, as you can see here in this image, is going to be the outer layer that surrounds the double layer serous membrane. You learned about the serous membrane in Bio 210. The parietal pericardium is going to be deep to the fibrous pericardium, and that's going to be the outer layer of the serous membrane. Okay. And then the visceral pericardium is the inner layer of the serous membrane, and it's going to be attached to the surface of the heart, also called the epicardium. So here, this is the walls of the heart. So there's three distinct layers. We have the epicardium, which is going to be the outer layer, and that's what's called, that's what's known as the very thin layer. Then we have the myocardium, which is the middle layer composed of cardiac muscle tissue, and this is going to be the thickest layer. It's going to do a big role of contraction. Then we have the endocardium, and the endocardium is going to be the inner layer, and that's going to form the inner linings of the heart chambers, and this is also going to be a thin layer. Okay. So the heart is divided into four chambers, as I said earlier. So the atria is going to be this thin wall upper chamber receiving blood returning to the heart. And the auricles, that's going to be on the outskirts, like right here, this is an auricle. So the auricles are these flat-like projections from the atria, which allows the atria to expand. And the ventricles are going to be these thick-walled lower chambers pumping blood into the arteries. So the right atrium is going to receive blood returning from the systemic circuit, from the superior and inferior vena cava and coronary sinus. And that's going to pump blood to the right ventricle. The right ventricle is going to receive the blood from the right atrium and pump blood to the lungs. And then the left atrium, as you can see here, okay, the left atrium is going to receive blood from the pulmonary veins and pump blood to the left ventricle. And then the left ventricle is going to receive blood from the left atrium and pump blood to the systemic circuit. Okay. So these are the valves of the heart. We have the tricuspid valve, the pulmonary valve, the mitral valve, and the aortic valve. Just know that the mitral valve is also known as the bicuspid valve. So what I like to do in remembering the position is I like to say try before you buy. So tricuspid comes before the bicuspid or mitral valve. So here the tricuspid is going to be located between the right atrium and right ventricle, whereas the bicuspid is going to be located between the left atrium and the left ventricle. So here you can see on the top left, the tricuspid valve. The top left is going to be the top left of this image. Sorry, this is the left. The top left of this image is going to be the tricuspid valve. The top right is going to be the mitral valve. And then in the center, you're going to see the aortic valve. And at the very bottom down here, this is actually the pulmonary valve. Okay. So bicuspid valve right here. So mitral bicuspid valve, tricuspid valve, aortic valve, and pulmonary valve. So here you have this rings of dense connective tissue that actually surrounds the origins of the pulmonary trunk and aorta and the orifices of the atria and ventricles. The reason why you have this is because this provides attachment for the heart valves and muscle fibers. And this prevents any excessive dilation of the heart chambers during a contraction. So these rings, along with other fibrous masses, in the interventricular septum actually makes up the skeleton of the heart. Okay. And so this is the blood flow through the heart. You can look at this and follow along. Okay. I don't need to really read this because it's right here. What I want to talk about is the blood supply to the heart, because just because the heart is gonna be the organ pumping blood to all the other surrounding organs like your brain, lungs, GI tract, and so forth. The heart itself is an organ, and so it needs to supply itself with blood. So the left and right coronary arteries is going to supply blood to the tissues of the heart, and the coronary arteries are the first two branches off the aorta, okay, or of the aorta. Oh, and so here, this is a chart, a nice diagram explaining the coronary arteries and how they supply parts, like how they break off the branches, into and how they like supply blood to the heart, and then the veins that collects the deoxygenated blood to go back into the right atrium. Okay, I think this is just a beautiful picture. And then last but not least, this side right here is the anterior side, while this figure is the posterior side of the heart, showing you just the identifying terms for the anatomical terms of the heart. So if you have any questions, let me know. You can definitely watch this video as many times as you need. If you think I went too fast, just slow it down. This is a PowerPoint, so you can definitely download it to watch the video that's embedded in here, as well as to enlarge any images that end up being a little bit too small for you to see. Okay, so stay safe, guys, and email me if you have any questions. Also, what I wanted to say is that you have assignments within your Connect within each week, so make sure you do those, and it goes and corresponds to what you've learned in this presentation. Okay.